Let's talk more about today and those clinical trials that you're referring to. So, cut to today, where are we with exactly what you just described? How did they come about? What are the advancements and what can we expect from the lab?
Yeah, great question. So in terms of where we are today, there's three trials that I want to highlight and just sort of like as examples of where we are today and what's possible for the future. The first trial is a trial that's currently being sponsored by CRISPR Therapeutics and Vertex, and what they're doing is they're using the CRISPR-Cas9 gene editing platform to knock out a gene called BCL11. And what that does is it restores fetal hemoglobin production. So that's important in a disease called beta thalassemia and sickle cell. So those are the two diseases where there's a need for hemoglobin production. And so by using CRISPR gene editing, you can restore hemoglobin production to enable, more normal red blood cell production. So CRISPR Therapeutics, in collaboration with Vertex, opened up the very first beta thal sickle cell trial about 3 years ago. And this year they've reported on some of the initial clinical data there. So we have the abstracts from the American Society of Hematology. They were published yesterday. And CRISPR Therapeutics disclosed that they've treated about 7 patients with either beta thal or sickle cell. And some of those patients have gone on to no longer require transfusions, which was the current standard of care. They seem to have a robust response in terms of restoring hemoglobin production through the editing of their stem cells using CRISPR-Cas9. So that's probably the most robust data set that we have today. About 7 patients in beta thalassemia and sickle cell, where it seems to really be making an impact. The other trial that I want to bring your attention to is a trial that was started by the investigators at the University of Pennsylvania. They published their results in Science Translational Medicine, and they're using CRISPR-Cas9 in the context of cancer. So remember, the first trial that I mentioned to you was within the context of a monogenic or an inherited disorder, sickle cell and beta thal. The investigators at UPenn used it within the context of cancer immunotherapy. So cancer immunotherapy is the concept there is that you use your own immune system and redirect it to attack the cancer. Now, what ends up happening is that a lot of times the cancer or the tumor will put up defense mechanisms to neutralize or to limit the efficacy of the cancer immunotherapy. So the investigators at the University of Pennsylvania in Philadelphia, led by Carl June, what they said is, what if we could use CRISPR-Cas gene editing to enhance the potency of the cancer immunotherapy, to enable the cells to overcome the barriers that are put up by the tumor, and that's what they've done. They've taken this cancer immunotherapy, which is directed towards a cancer called synovial sarcoma, which is a cancer of the bone, and they've taken the T cells that attack that, and they've enhanced them. They've souped them up by genetically modifying the cells with CRISPR-Cas9. They've treated about 3 patients, and the responses look really good there as well. So that's the second trial that I think is worth highlighting, where we have some really robust clinical data. And then the third trial is, within the context of HIV. We know that some people are not susceptible to HIV infection because they have a mutation in a gene called CCR5. So these folks are naturally immune to HIV infection. So the idea here is, what if you could use CRISPR-Cas9 to knock out the CCR5 gene and eliminate HIV from infected individuals? So this trial that I'm referring to was actually carried out in China. It was a case report published in the New England Journal of Medicine earlier last year, where they took a stem cell, knocked out CCR5 which is the gene that HIV uses to enter into a cell. So they knocked it out so that HIV can no longer enter into that cell. So they knock out CCR5 from the hematopoietic stem cell, and then they reconstitute the patient's immune system with the CCR5 knockout hematopoietic stem cell. And the results have been really interesting. I mean, it's safe, it's effective. And so what I've highlighted for you is three cases, one in an inherited disorder like sickle cell, another in cancer like synovial sarcoma, and the third in infectious disease, where we have some promising clinical data that shows that CRISPR gene editing can be used to lead to potentially therapeutic beneficial outcomes. And so to answer your question, I think the future of CRISPR therapies is going to be built on the foundation that's been established by these. So we're going to see more in the arena of inherited disorders. We're going to see more in the arena of cancer immunotherapy, and we're going to see more in the arena of infectious diseases.